Thank you to the organizers of this meeting, and thank you for coming. So, genome editing technologies are viewed as fundamentally distinct from other therapeutic strategies like small molecules. And our work is seeking to determine how small molecules could be used in combination with genome editing technologies, and whether modulation of genome editing technologies with small molecules could provide a means for fine-tuning the effect of genome editing. We're also interested in how genome editing technologies might have unexpected interactions with other small molecules or drugs, which might therefore impact the therapeutic impact of genome editing technologies. So in this talk, I'll focus on two areas. One, are there any similarities between the way cells respond to genome editing technologies and the way they respond to small molecules? And if so, what are the possible pharmacological implications of the interactions between genome editing technologies and small molecules? So we know that small molecules and gene editing technologies, or indeed many other forms of gene therapy, is that they are very different, and in fact we tend to study them in silos. So small molecules, of course, are very low molecular weight, so they are easy to, they can easily enter cells. In contrast to genome editing technologies and other forms of gene therapy that are composed of large molecules that are very hard to deliver inside cells. Small molecules and gene editing technologies also differ in terms of how they target specific biological processes. So we know many small molecules target specific proteins, while gene editing technologies are geared towards editing specific DNA sequences. So are there any similarities between the way cells respond to these two types of therapeutic strategies? And in particular, I'll focus on genome editing by CRISPR-Cas9 systems. And so for CRISPR-Cas9, there are two main components to think about. So one is CRISPR guide RNAs, which guide the Cas9 endonuclease to target specific segments of DNA that you may want to edit. But because CRISPR-Cas9 is a big molecule, you need different ways to deliver the technology inside of cells. So commonly, electroporation is a simple means of delivering DNA and other components like CRISPR-Cas9 into cells. And normally, you can deliver CRISPR-Cas9 either in form of RNA, whereby the the Cas9 mRNA delivered into cells can then be translated into the Cas9 protein. There are other means of delivering these components, and the problem is that depending on what delivery method you pick, you might get different efficiencies of genome editing, and also there might be toxicities associated with each of these delivery methods. So if we look into a little bit more detail of how cells might get perturbed by genome editing technologies and how you deliver the genome editing components. So let's take the instance where you're delivering Cas9 mRNA and a guide RNA sequence through electroporation. So one thing, electroporation in itself might actually perturb the cells in multiple ways. So for instance, electroporation can cause apoptosis. Of course, when electroporation changes the permeability of the cell membrane, it could also perturb many processes inside cells. And once the mRNA gets inside the cell, this mRNA might actually be detected by cellular mechanisms of antiviral defense, and as a result, you might have activation of antiviral defenses inside of cells that can destroy the RNA that you delivered, but it also activates several downstream processes that might affect how much editing efficiency you get. Furthermore, when CRISPR acts to edit your DNA sequence, one of the challenges is that CRISPR cleaves the DNA, but it's the endogenous repair system that kicks in to repair the DNA that you've cleaved. And there's a problem with that because many times the repair occurs by non-homologous end joining that is highly error-prone. And these events can lead to activation of DNA damage responses inside the cell. The other option of delivering Cas9, CRISPR-Cas9 into cells is using a Cas9 protein and electroporating in the presence of guide RNA. And that also will still activate downstream processes in the cell that are independent of the target DNA sequence that you're trying to edit. Another common delivery method is using viral vectors, so like V6. And this kind of delivery that has been used for other gene therapies also will stimulate some genotoxic events inside of cells. And all of this is going to determine how efficient your genome editing is attained. So in a recent study, it was shown that basically the delivery of CRISPR-Cas9 components into cells actually activates or induces a large amount of transcriptional responses towards the genome editing components. And in particular, delivery of Cas9 mRNA cells was associated with very strong antiviral transcriptional responses inside of cells. So you can see that there are a number of genes associated with viral response inside cells that get induced when you deliver Cas9 mRNA inside of cells. There's also a lot of stress response that is seen, as well as DNA damage response and cell cycle response. So you can imagine what impact this can have on the editing efficiency that you get. And so because of all of those transcriptional responses, it's very much possible that other small molecules might induce similar transcriptional responses to the responses we see with the CRISPR-Cas9 components. So in one of our studies, we are using the connectivity map database that is basically a database that now has about 1.3 million profiles of small molecule perturbations across a range of cancer cell lines. So our idea basically is to map the similarity between transcriptional responses to small molecules versus transcriptional responses to genome editing components. So previously, CRISPR-Cas9 has been used. Previously, the connectivity map database has been used to match transcriptional responses of different biological states to transcriptional responses from small molecule perturbations. But in our case, we are trying to match how gene editing components might induce similar transcriptional responses as small molecules in the connectivity map database. So you can imagine a case where if you take the perturbation signature from genome editing technologies and match it to the contents of the CMAP database, you might get a match in which genes that are up and downregulated by the gene editing component are also perturbed in a similar manner by a specific small molecule. So when we do that, we find that there are a number of small molecules that actually show very strong patterns across several cell lines that are similar to patterns that are obtained by perturbations by small molecules. And interestingly, many of these small molecules target several processes involved in DNA repair. And this is very much consistent to a recent study that has shown that several DNA repair targeting small molecules can actually impact genome editing efficiency by CRISPR-Cas9. And we're also investigating the role of the structures of these small molecules. So we see, for instance, that many of these molecules have similar chemical structures that could underlie the responses observed. We've also identified several small molecules that show perturbation signatures that are the reverse of specific small molecules. So you'll find that some small molecules will downregulate the same set of genes that are upregulated by some CRISPR-Cas9 components, and there's several of them that we've identified. And among these molecules, interestingly, we find adrenergic receptor agonists as enriched in the small molecules that show that signature, and this is consistent also with some recent studies that performed high-throughput molecular screen to find small molecules that might impact genome editing efficiencies. So you can begin to imagine what kind of implications this could have at the therapeutic level of genome editing. And therapeutically, genome editing technologies can be delivered either in vivo or ex vivo, in which you take patient cells, modify them in the lab, and then inject them back into the patient. So one implication could be that basically you could use these small molecules to intentionally modulate the efficiency of genome editing. And you could also use, and potentially, the fact that some small molecules can interact with genome editing technologies means that you can have unintended side effects of a small molecule that is present when you're delivering the genome editing technology. I'll just highlight some limitations of our current study that we're trying to address. So one is that the data we used in this case was from perturbation of CD34 positive hematopoietic stem cells, and there's need to replicate these in other cell types. But in conclusion, we see that this approach could be extended to identify several other small molecules that can enhance or inhibit genome editing. And indeed, Sigma is already selling some a small set of small molecules obtained from literature that can enhance genome editing. But we think the approach of using transcriptional data could help us to understand the mechanisms underlying these responses. So you can imagine a future in which small molecules can be intentionally combined with genome editing technologies to obtain very fine-tuned responses to genome editing. And furthermore, it will be important to understand how drugs and genome editing technologies can interact, because this could have therapeutic impact in toxicities to genome editing technologies. Thank you.
Questions for Geoffrey? Okay, I have a quick question. So in terms of the method that you presented here, it was going to connectivity map with the signature of the CRISPR-Cas impact, and then finding a small molecule with the strongest match. Now, the connectivity map just ranks profiles and links, which are by the score. It doesn't have any further it doesn't necessarily tell you how strong the match of the signatures is, and whether the top one is just a lousy match or a really good a very similar profile, right? It's just the best match. So
yeah, you can compute some enrichment scores, which can be based on...
Could you use the mic?
Yes, so you can basically rely on enrichment scores that are basically comparing your signature versus a random match.
Right, but a significant enrichment, would that be enough to tell you that this will improve the efficiency of the CRISPR?
So that is an interesting question that we are trying to work out. So we know, for instance, from some of the molecules that we've identified, that they've been actually been shown to have an impact on editing efficiency. There are others that haven't been shown, and what we think is that not all of the transcriptional responses are relevant to the editing efficiency. However, they might be relevant for other things like toxicity.
Mm-hmm. Okay. Thanks very much. Let's thank Geoffrey once again.
Wait.
Oh, one more question.
Yeah, I have a question that's more about the biology of your findings. So do you know what the biological basis of topoisomerase inhibitors and HDAC inhibitors are for, having an impact on this?
So I can't say we know for sure, but it's definitely connected to the fact that CRISPR will cleave the DNA, and that initiates some genotoxic response.
Yeah, but you would have thought that the topoisomerase inhibitors would actually impede that because they're important for repair.
Yes, but it's a combination between a genotoxic response that leads to apoptosis versus one that leads to actual repair. So if you get apoptosis, then your cells are dying, so your editing efficiency goes down basically because the cells are dying. So.
Yeah, but the cells could die from too much damage, right?
Yes.
So if you put in a topoisomerase inhibitor, isn't that going to make it worse?
Yes, in principle it would make it worse, but yeah.
Okay, so and also the HDAC inhibitors, do you know what the basis for that is?
So in the paper that was just published through a molecular screen, they try to propose a mechanism, but I don't think we can say that that's what happens for sure. Like, yeah.
The speaker once again. Are you Gregory?